All right, so I'm going to start with a snapshot of universal cells. We're a Seattle-based biotech company founded in 2013 by myself and my co-founder, Dr. David Russell from the University of Washington. And our goal is to deliver stem cell therapies that overcome immune rejection and also to bring off-the-shelf cell therapies to the market. So our technology is based on our proprietary recombinant AAV platform, gene editing platform, and we use it to to modify the HLA molecules in the stem cells so they become universal donor cells. So because this is a very broad platform, we leverage the multiple opportunities through our dual business model. On one hand, we establish partnerships with other companies in the sector to enable them to bring these universal donor cell stem cell therapies to the market, while we are also developing our own internal products in immuno-oncology. So since 2013, we've had quite a lot of progress. We've built our in-house vector and cell manufacturing. We're about to start to build our own GMP facilities that will enable us to produce our own products for clinical development for phase 1 and phase 2. This photo is already outdated. We are now with 25 employees as of today at the company. And we've done all that mostly through self-funding ourselves with our revenues from our partnerships. We haven't, as of yet, had to raise institutional investment, and we've been cash flow positive for the last couple of years. So Universal Cells is developing off-the-shelf cell therapy products that avoid a main hurdle in stem cell therapies, which is the rejection by the immune system. So we create gene-edited pluripotent stem cells, universal donor pluripotent stem cells that can be indefinitely expanded. Then they can then go through well-defined differentiation processes to create many different therapeutic products that are then administered as a high-quality, low-cost cell therapy that can treat many unmet medical needs. So with this approach, we have thoroughly characterized materials and processes that, combined with the possibility of having a large-scale manufacturing and distribution, and we avoid the use of immune suppressants. So we think that this altogether results in improved clinical trial designs and much better patient outcomes with much lower costs. So to create these universal donor cells, we use our recombinant AAV gene editing platform, which we think is the safest method to edit the genome that is available. Because we don't have any nucleases, we don't promote any genotoxicity, no off-target cuttings, and no on-target mistakes. The vector in itself is the delivery system. They are very efficient in transducing stem cells. And finally, these vectors have already been clinically validated and tested in humans, so that offers a straightforward regulatory path and makes it an ideal manufacturing reagent. So to control HLA expression, we engineer both HLA class 1 and class 2 because these have been shown to be the most important molecules involved in rejection of cells. So for class 1, what we do is knock in a non-polymorphic HLA gene at the beta-2 microglobulin locus to replace all class 1 molecules by a decoy molecule to avoid the lysis by NK cells. For class 2, there is no drawback in having no class 2 molecules on the surface, so what we do is we remove all class 2 molecules by knocking out a transcription factor that is important for the expression of class 2 molecules. So in the end, we have a universal donor stem cells that is engineered both for class 1 and class 2. So to recap the universal donor cell editing strategy, we start with an unedited iPS or ES cell that we engineer both class 1 and class 2 genes, and we end up with a stem cell that expresses only HLA-E, which is a non-polymorphic HLA on the surface. And we can also, during this process, do other additional editings to the cell if desired for the final product. For example, during the process of engineering the HLA molecules, we can introduce suicide genes to enhance safety profile of the final product. So once we have the universal donor stem cell, we can then produce the therapeutic cells through different differentiation processes and obtain the final product, which is the universal donor differentiated cell. So we've shown extensive preclinical proof of concept that our cells are not recognized as foreign. We've shown that they don't trigger an immune response neither in vitro nor in vivo. They do not. They're not targeted by T cells. They're not lysed by natural killer cells, and they're not recognized by human anti-HLA molecules. And we've recently published our latest results in Nature Biotechnology in May this year. So we think that these cells have great potential for clinical use, and especially because they retain, even after gene editing, they retain the parental cell characteristics. They retain full ability to differentiate into the different cell types. They do not show any toxicity resulting from the gene editing that has been done, so we think that it's a great cell for clinical use. So going back to the business model that I talked about, we have two sides of it. One is the partnership programs, and also our internal therapeutic program. So for our partnership programs, we look for companies that have complementary technologies and expertise. We don't want to reinvent the wheel, so we partner with the best in the field, and we are looking for potential for a faster path to the clinic and the validation of these cells in the human and getting to a final product and an approved product. So far, we have partnered with 3 companies. Our first partnership was with Adaptimmune to create universal donor T cells. We also partnered with Helios to create universal donor RPE cells, and also for universal donor liver and kidney organ buds. And just 2 days ago, we announced a partnership with Blue Rock to provide universal donor cells for their platform. On our internal therapeutic program side, we are engineering universal donor natural killer cells to treat cancer. And we are designing our product to address the main challenges that are currently preventing the widespread use of these cell-based therapies to treat cancer. And so we believe that we have several advantages. Our universal donor natural killer cells have several advantages over allogeneic NK cells. The first one is, and a major one, is that they do not elicit immune rejection nor trigger the development of anti-HLA antibodies. These cells can be engineered to have an enhanced tumorigenic activity via our gene editing technology. Having a well-characterized product, product that can be used, eliminates donor variability. That is a big problem in natural killer allogeneic therapies. These cells are derived from iPS cells, so they are immortal. We can scale them up indefinitely, so that brings the cost of goods really down. We can treat many patients with these cells. And the last point is, because these cells are not recognized by the immune system, and and do not trigger the development of anti-HLA antibodies, we have the possibility to redose if necessary. So as a company, we hope to have several value-creating opportunities in the next few years, both through our partner and our internal programs. The milestones that we are expecting to achieve in the next 3 to 5 years from our partner programs are, we hope to have the first human proof of concept of our universal donor cells in one of our partner programs, and we hope to have at least 2 other partner programs in the clinic within this time frame. And as of for our universal donor NK cells, we also hope within that time frame to have the first results from our phase 1 clinical trial in our first oncology indication, and we hope to have a second IND filed by then. So I'm going to leave you with a summary of universal cells. We have this one universal donor cell that can be the basis for many, many therapeutic products. As a fan of the Lord of the Rings, I like to say one cell to rule them all. And we have this very broad universal donor cell platform. We have been self-funded through revenues from partnerships, and we have reinvested these revenues in our own internal therapeutic program that we're developing in immuno-oncology. Thank you very much.